All right, perfect. So let me also do a screen share, and then you guys should be able to see my screen. I'll bring the PowerPoint in the forefront here. Okay, so as, uh-oh, what happened there? You guys see my PowerPoint screen? Or is it you actually see my Finder? I
We
might
see
not.
your Finder.
Oh, okay, this is interesting. Let me quit my share and then I'll reshare. It's so strange. It's as if PowerPoint just kind of disappeared. Oh, I guess it accidentally closed the window. There it is. Okay. Sorry about that. Already having technical difficulties. Okay, so as Bridget mentioned, my name is Jack Panopoulos, and my email here is jack.flojo@bd.com. So if you guys have any questions that arise during today's session, by all means, you can either use the chat box today, you can open up your mic maybe at the end and ask question verbally, or you can save your question and send it via email if you'd like to send data or screenshots of things that you are experiencing. Okay, so let me just open up Flojo here, and today's session is the intro to Flojo session. So I'm going to be covering a lot of sort of basic manipulations in Flojo, show you how to annotate your data, for example, how to do a little bit of compensation, and also how to work with the layout editor to get a lot of plots kind of in the format that you would like for making figures for a paper, for example. Also, we're going to talk about how to export statistics from the table editor so that you can get statistics for your manuscripts as well. Okay, so the first thing that you need to do in Flojo, generally speaking, you can see that it says drag samples here. The nice shortcut in Flojo is you can actually take the folder that contains the actual FCS files. So if I actually look inside this folder, you can see that the data files have this extension .fcs, and that stands for flow cytometry standard. Flojo will also read CSV files. It'll read LMD files that come from Beckman-Coulter type machines. There's also Miltenyi makes a max quant, and it has the extension MQD. Flojo will read those files just fine, but I always recommend people, if you have the chance, make sure that you export your FCS or your flow cytometry files as FCS, because that's the format that Flojo works best with, and you'll experience, fewer sort of graphical problems if you export in that format. Okay, so we're just going to take the folder that contains all of those files and drop it into the workspace, and you will notice that the bottom section here will list out all of your FCS files, and then you'll notice kind of at the top or sort of in the middle pane here, there's an all samples sort of ledger, and then there's a compensation ledger. When I dropped the immunophenotype folder into the workspace here, it actually created the immunophenotype data basic folder as well. So this ledger got added because we dragged in a folder. If you drag in the individual FCS files, this would not be here. Okay, but this middle section is known as our groups pane, and we use this as an area to stage files. So if you want to separate them based on whatever experiment that you have, perhaps you want to separate your files based on time point or drug concentration or something like that, you can segregate your samples here in the groups pane by using this little tool at the top with the bracketed plus. And I'll show you how to use that momentarily. Okay, but by default, Flojo is always going to have an all samples group, which is going to contain all of the samples in a given workspace. And then the compensation group is always going to be present as well. This group actively looks for any files that are labeled comp or unstained. Okay, I'm going to go ahead and just save this real quick to the desktop. All right, and then at the very top of the workspace, there are several functions that are quite useful. So the black ball here where the pyramid is inside has a lot of shortcuts, like opening a new workspace, right? Creating a new one, opening one that you had already saved, and of course, saving workspaces as well. You have the undo redo buttons right next to the black ball, and then at the very top here is the taskbar with some very important buttons like adding samples. So if you don't want to drag and drop, you can always click on the tube with the plus and navigate through your file system to find your data files. Okay, so it's up to you which method you want to use. I think drag and drop is a lot easier, so I seldom use this functionality. Okay, and then the table editor and layout editor, these two buttons you'll see me push a little bit later because they are major features in the Flojo program that will allow you to export data for your manuscripts. So we'll get into those a little bit later as well. Just below the taskbar is our band ribbon interface. So you can see that there's a bunch of tabs here at the top. I don't think it's abundantly obvious that that's what they are, but if you click on them, you'll notice that the ribbon underneath kind of changes as you click on the different tabs. I'm going to try and show you the most efficient way to work through your data so that we're not clicking around through all of these tools and tabs throughout the entire session. So, you know, my motto is kind of like, you have the data in hand, now you want to like get the data analyzed as fast as possible and then, you know, go outside and have fun or something. So once we have the data files in hand, I like to sort the data, you know, if necessary, just so that I can see things kind of either in alphabetical order or perhaps based on, you know, some other information that's contained within the file. I can talk about that when we get to the metadata step in a second. But when I bring the data files into the workspace, you'll notice that there's three icons that sit to the left of the files, right? You have this sort of diamond shaped icon, then you have a circle, and then you have this open square. You will immediately notice that some of my files have this open square, others have this gray grid. Okay, the gray grid means that these files have a compensation matrix attached to them. So if I roll my mouse over the gray grid itself, you'll see that it says compensation matrix in bold. And then after the colon, it says acquisition defined. So what this is telling me is that these files have been compensated, and in this case, they happen to be compensated on the machine, on the instrument that I ran the samples on. Okay, so technically speaking, these are already compensated files. I probably don't have to do any compensation on them, but it's always a good idea to inspect the compensation matrix that is sitting on your files, regardless of where you do the compensation, because having poorly compensated files in a data set is going to lead to erroneous statistics in your reports later on. So it's always a good idea that before you do any kind of gating or anything to inspect the comp matrix. So that's what I'm going to do here. I always do a little bit of QC on the data first before analysis, and that includes looking at the compensation matrix. Now, what I like to do is I like to preview that compensation matrix on a fully stained file so that I can see all of the colors and see how does the compensation look on the actual cells that I'm going to analyze. And so what I'm going to do here, I double clicked on that gray grid, and that will open up this window that we're looking at right now. This is called the matrix editor window. And I'm going to enlarge it here so it's easier for you guys to see. Okay, in this window, there are three panes. The upper left has the list of compensation matrices that you have in the data workspace. So in our case, we only have one right now, and it's this one that came from the machine, right? The acquisition defined matrix. Then in the second pane on the right-hand side here, you have a table with all the fluorochromes and the various spillover values here that they have into the different detectors that are present in the data set. Now, normally I don't pay too much attention to the table values because, again, it's more about inspecting the visual here and understanding what is going on with the compensation. So for that, I normally pay attention to this series of plots that you see down below. We call this entirety of these plots an n by n plot because what is being presented here is every 2x2 color combination that is present in the data set. And so we have an 8 or 9 color data set that we're going to be showing today, and every 2x2 color combination is being shown here. So I'm going to go ahead and zoom in just so it's easier for you guys to see. Each of these plots shows 2 different colored dots. So you have blue dots and then you have black dots. Okay? The blue dots represent the data as they are without any compensation. So if you ran the data and then you were going to begin to analyze the data without having any compensation applied to it, you would see a lot of strange looking populations like the one we see here kind of in the middle plot, right? Where you have these sort of diagonal populations that look to be double positive for whatever these 2 colors happen to be. Okay? This is a telltale sign of not having done compensation. Okay. Now, again, I mentioned we did compensation on the machine, and that's what's being carried forward onto these files. The compensated data, or how the cells look now with the compensation matrix applied, are the black dots. So normally what I do here is I turn off the overlay uncompensated, right? So that we can actually see what the current data look like. And so normally I'll start over here in, one of the corners. And what I'm going to look for in a lot of these plots is I want to make sure that these single positive populations that you see here, either along the y-axis or along the x-axis, I want to make sure that they're relatively parallel to either the y or the x-axis. I don't want to see bending of these populations, crashing in towards the y-axis or down towards the x-axis. I also don't want to see banana-shaped populations that are bending towards the center of the plot. Okay, so if you want to get technical about it, I want to make sure that the median of the single positive population on either the x or the y axes matches the median of the double negative population that you see in the corner, in the sort of bottom left corner here. So, you basically want to take a ruler and be able to, show that the median here matches the median over here. Same thing on this side, the cells that are really, furthest out to the right, are parallel or in the middle with respect to the double negative population on the opposite side. Okay, so as I scroll across, I can see a little bit of minor bending on some of these plots. So for example, here we have a little bit of bending downwards, but it's not, it's not severe. And the vast majority of the population looks to be pretty parallel to the x-axis. So it looks good. This plot in the middle, right, also looks pretty good with respect to the populations. Don't be afraid if these single positive populations begin to fan out a little bit. They sometimes form like trumpet-shaped populations, or dumbbell-shaped populations. That's okay, so long as the dumbbell or the trumpet is sort of evenly distributed across the negative and positive, and that the middle of that trumpet, again, matches the middle of the double negative population. Okay, so these data, here's a trumpet-shaped population that you can kind of see sort of forming a funnel as it gets brighter. But again, I'm looking at making sure that the middle of this population is matching the middle of the double negative. Okay, so I'm going to say that most of these data look pretty good, and that's not a great example, to show you if I'm trying to show you something about data quality control. It's a good example of what is good, but what does a bad matrix look like? To answer the question, there was a question about, will this be recorded? Yes, I will. It's currently being recorded, and what's going to happen is after the session today, it'll take about 30 minutes to an hour for the link to be compiled. I will send it to Bridget, and then she can distribute it to you guys. Alternatively, if you guys want to ping me directly, you can send me an email and I can send you the recording. Okay, so back to our workspace here. What I want to do is I want to show you an example of a poorly compensated workspace. So this is a different experiment, different colors, but we'll show you the example of something that is poorly compensated, that's something that's done really, really badly. Okay, so here, this experiment's only 4 or 5 colors, and you know, when we look at this matrix, let me zoom in a little bit here, and I'll go to the second row because this is probably the most obvious. But if we look at the second row here, you can definitely see that this population is hooking right towards the left. Looks like it's going to crash into the y-axis. Looks pretty askew. Right, same thing over here. We can see that this population is starting to fan out, which is fine, but you can see that it's fanning out and it is skewed towards the left. It's not evenly distributed across the middle so that the middle of this population matches the middle of the double negative. If we go down a couple notches here, you can see that there's a population stretching down towards the x-axis, another one here that's reaching and looks like it's going to crash into the y-axis. A little less obvious is if we look at these plots on the left, we have a nice round single positive population here, a nice round single positive population here. But if we, walk these populations back, again, looking at the middle of these populations and walking them back towards the double negative, right, where they sort of intersect here at this population in the, kind of in the center of the plot. If this happens to be the double negative that I'm, waving my little mouse around, one has to beg the question then, what is this really weird population that is even more negative than that off towards the left, right? So again, because the medians of the single positive populations here are askew of the double negative, we know that something is wrong with the compensation, in addition to the hooking that we can see over here. Okay, so bad compensation, the question then becomes, why did this happen? What did these people do that caused this to happen in the first place? Okay, and so in order to sort of begin troubleshooting that, we have to kind of remember the 3 rules for good compensation. And these come from Mario Roeder. Many years ago, he published a paper kind of outlining these rules. And for the most part, they have remained unchanged for the last, I don't know, I'd say probably 25, maybe 25 years, something around there, 20 to 25 years. We only started to get to multicolor flow, like 4 colors around, I want to say 95 or so. But in any case, here they are. Okay, so rule number 1 is making sure that your compensation controls, okay, the single stain controls need to be at least as bright or brighter, right, than any of the samples that you wish to test. Okay, so that's an important rule to remember. And I have to say that when I look at people's data that have compensation problems, 70% of the time it's because this rule is violated that the compensation doesn't work out properly. Okay. So rule number 2 is background fluorescence must be the same for positive and negative control. This is pretty straightforward. If you have a mixture of compensation controls where you're using, let's say, cells for a viability dye, but you're using beads for all the other colors, you just have to make sure that you have unstained cells, right? Cells that have not been exposed to the viability dye as a negative for that cellular stain. And then you also have to have unstained beads for all of the bead colors. Now, normally manufacturers include a negative bead in the positively stained tube. So you normally don't have to worry about carrying around unstained beads. But when you have a mixture of cells and beads, it just means that you have to match cells and beads to each other. And we'll do this in today's session towards the end with compensation. Okay, and then the third rule is making sure that the compensation controls emission spectra, right, or whatever the fluorochrome happens to be. The fluorochromes must match each other. So if you're using Alexa 488, in the experiment, then your single stain control also must be Alexa 488. You can't use FITSI, or God forbid, something like GFP, right? Even though these fluorochromes all have very similar emission spectra, they do differ and they will cause problems when it comes to applying or calculating the compensation. So making sure that they match is very important. All right, so those three rules in hand. I should mention a fourth rule, which is kind of the ghost rule in a sense, is that you want to make sure that your panel design is also solid. So panel design is a discussion in and of itself, but you have to make sure that the color panel that you wish to use on your cells is compatible with the instrument that you are going to run those samples on. You could have a beautiful panel that has separated, colors, very nicely separated colors, but if the instrument that you intend to run it on doesn't have, a detector for one or two of those colors, or is not optimal for those colors, then you're going to have problems as well. So panel design, which comes first, is also very important to making sure that your compensation, works out in the end. Okay, cool. And Bridget says there's going to be a panel design workshop on October 12th. So that's coming pretty soon. And I would highly recommend, especially those of you that are brand new to flow, even if you've been in flow cytometry for a while, I think it's always nice to have a refresher on panel design, because number one, you can ask questions about new dyes that are out there and how, how these dyes can work in spectral machines, et cetera. So there's always new stuff coming out, new things to be aware of. And I highly, highly recommend that you take a panel design course. Okay, so those are the rules of good compensation. Now let's go back to the workspace and figure out what the heck went wrong here, right? So all of these problems, which one of the rules did they violate? So normally the first thing that I will do if I see, compensation trouble, the first troubleshooting test that I will do, and pretty much the only one that you can do that is going to be immediately obvious, is to overlay the fully stained sample, right, with each one of your compensation controls. And so I've done that for you ahead of time here in the layout editor. So you just take each single stain control, you overlay it with the fully stained sample, and I like to look at it as a histogram, but you look at it along the uncompensated parameters. So you have the, in this case, we're looking at BV421 along the x-axis. So the compensation control that we're including here is the BV421 compensation control. And then the sample is whichever one. Generally speaking, you want to pick the sample that you expect to have the brightest cells in. So if you know that when you treat the cells with a particular drug that they're going to increase expression of X, Y, or Z, you probably want to look or overlay that population or that sample with the appropriate single stain control. Okay, so in each one of these plots, the single stain control is in red, the sample is in blue, and then the x-axis is going to change based on whatever color that single stain control happens to be. Okay, so in this case, you can see that the blue stain, fully stained sample, right, the brightest cells fall within the range of the brightest single stain control peak here. So as far as compensation is concerned, BV421 was fine. Okay, if we move over here to BUV 395, you can see that the cells and the compensation control are as bright as each other. So this is also okay. There's nothing wrong with this type of a situation. Okay, so we're okay in this regard as well. So BUV 395 passes the test. Okay, but look what happens here when we get to BV 605, right? Suddenly, we can see that the blue sample, right, some of the sample is actually brighter than the single stain control. So this is a problem. Okay, so BV 605, compensation control in this case, just from a technical standpoint, we'll talk about this, another problem going on here in a moment, but from a technical standpoint, it is not bright enough to cover the brightest peak that you have present in the data set. Okay, so that's going to cause compensation issues. If we go over to PE, the same thing, right? You can see that the compensation control peak is here, but the cells are all the way out here at 10 to the fourth. Okay, so there's two compensation controls now that have problems. We know that's definitely going to lead to compensation errors. Okay, and Alexa 647 is fine. The cells are not particularly bright in that channel anyway. Now, with respect to PE, PE is one of the brightest fluorochromes that you can use for flow cytometry. So I'm assuming here for this compensation control, they picked a surrogate marker that was not very highly expressed on these cells. And therefore they got a really poor comp control. I would pick something that's fairly abundant on the cell surface, especially if you're looking at lymphocytes. You might choose something like CD4. But in this case, they didn't, or maybe CD45, something along those lines. Over here, when we look at BB605, even though technically the comp control is not bright enough, what I'm actually gaining from, what I'm understanding from this particular plot is that the background fluorescence, right, of the sample is quite high. It's out here 10 to the third. Background fluorescence, the comp control is around 0. Why is this, 3 orders of magnitude higher than, what the background ought to be? Most likely what happened in this experiment is that these folks did not titrate the antibody. So I assume here, if they had titrated the antibody, maybe 3-fold, 4-fold, something like that, that would bring this, negative down, but it would also have the consequence of bringing the positive down into something that was reasonable, and this comp control probably would have been all right. This is a very bright compensation control, and I would normally say that this is, would be pretty good. But again, titration, they're wasting antibody, and the comp control now is not going to work in their system. Again, here in this one, even though the comp control is okay, you can kind of see that they didn't titrate the antibodies. They can definitely bring that background down a bit, and so again, they're just wasting antibody. Same thing over here. Okay, so always important, if you're brand new to flow, always titrate the antibodies, for the experiment that you're going to run, so that you know what the optimal concentration is going to be. Not only is that going to save you some money, but it's also going to avert these types of problems where you just completely blow the brightness of your compensation control out of the water. Okay, so very important: titration, panel design, compensation controls. All right, so that's the reason why these guys' experiment failed. And I will tell you that, I would just say rerun this experiment, redo it using the proper titration first. So titrate out the antibodies first, and then I would tell them to find another surrogate marker for PE, because this is clearly not bright enough for the sample that you are trying to analyze. Okay, so wasted time, wasted money with that experiment. All right, so now let's go back to our original experiment here. So that's data quality control step number 1. QC step number 2 is just to open up the data file and look for clogs or air bubbles in the data set. The easiest way to do this is just to, double click on a file. Generally, I'm going to look at everything except my compensation controls. But you would change the x-axis here over to time, and then the y-axis, it's generally a good rule of thumb to put this on a parameter that's more of a sort of a far red emitting dye, because the red laser has a little bit more wobble to it, and it's often easier to see anomalies in the data when you look at them off the red any color that's sort of excited off the red laser. So here, what we're looking for is to make sure that all of the events, right, all the dots are sort of evenly distributed from left to right across the screen. Okay, not top to bottom, but left to right. So does the right side of the graph look similar to the left side of the graph? Yes or no? And then just, use the little arrow keys here on the right, making sure that you can see that there aren't any major anomalies. Now, again, these data are pretty clean, so there's nothing to show here. but I do have an example of a workspace that has an anomaly associated with it. Okay, so this data file, if we look at it here, we've got time along the x-axis, we have Alexa 647 on the y, and here you can clearly see that there's a bit of picket fencing or like these striations going through the data. It sort of kind of looks kind of weird in the beginning, like up to the, maybe the 20-second mark or so. But then after that, it looks pretty smooth, right? Everything looks nice and evenly distributed. Now, the reason that we want to chop out clogs, that's what's happening here. The reason that we want to look for these anomalies is because, oftentimes when the machine clogs, you'll get data points like this, where they get shifted downward relative to the rest of the sample, right? Alternatively, they will also get shifted up in an upward direction, right? So you may find a new, really bright, rare population of cells in your data set, but you can clearly see here that that rare population of cells might be due to the fact that they're just, a spike. So for example, here, right, these really bright Alexa 647 cells, this cluster that you see right here is really just an artifact due to the fluidic pressure changing. Okay, so we want to be aware of this, and we want to be able to clean this, remove it from our data. So normally what I tell people to do is either just create a gate around the good stuff, right, to exclude the other stuff, maybe call it time or something like that. And then when you double click on this, you'll carry over only the good data. There are some tools that will help you to automate this. So I like to talk about Flow AI. It's my favorite algorithm right now for cleaning data up, although we just released a new one called Peacock. I have yet to try that one, but Peacock is supposed to be an improvement upon Flow AI. But I think this in and of itself is a great algorithm. So in any case, you can run Flow AI on the data set or on an individual file. And what it's going to do is it's going to generate two populations, a bad events and a good events population. And let me juxtapose those two so that you can see them right next to one another. I made the good events a little bit larger than the bad events, but you can see here that the algorithm, once it finished, right, it cuts out the bad data here, leaves you with the good stuff, and you can go ahead and gate your samples accordingly. Now, I saw momentarily here that there was a hand raised. Please use the chat box if you guys have a question. The chat box should be at the bottom of your screen, and you can enter a question there. I'll stop, look at the question, and then answer it. All right, so that's something to look out for in the data set as well. Okay, so the if there's any clogs or spikes in the data, you want to gate those out. You can use either manual gates or you can use the Flow AI tool. Okay, if you don't have Flow AI, I'll just show you where you can get it. So to get Flow AI, you would go to the Workspace tab, go to the Plugins drop-down menu, and click the top option that says FlowJo Exchange. Okay, this takes you to our website that hosts all of these plugins. There you can see our Flow AI. You just download this and follow the instructions that are included in the zipped file to install that particular tool. Okay, there's a lot of other tools here that we'll talk about next week, but that one in particular I think is quite useful, especially if you're running plates and trying to do high throughput type stuff. Let's see, there's a question here. What could be the cause of the bar gets thicker after time? Is that just if we change the uptake sample from low to medium? Yes. So if you change the speed of the acquisition from low to medium or low to high, it'll look less dense in the beginning of your acquisition, and then after you hit the medium or high button, all of a sudden it'll look a heck of a lot more dense. For any acquisitions that occur like that, you probably want to not use Flow AI, or at least dampen Flow AI's sensitivity, because it looks for changes in fluidic pressure, and when you change those buttons, it obviously changes the fluidic pressure so that the cells go faster through the stream. I know that Peacock is a bit more tolerable of such things, so you may want to try that algorithm. And as I mentioned, Peacock was just released, so I think it's down here at the bottom. There's also a new one, Fate, but there it is, Peacock, another quality control algorithm for cleaning up the data, due to data spikes like air bubbles and such. Okay, so QC is done. Next thing that we want to make sure of is annotating the data. Okay, now in my case, there's two areas that you want to include for annotation. Number one, look at your parameter list. So double click on an FCS file, preferably one that's fully stained, one that you know you're going to be using to create figures and things. And then expand your drop-down menu. It doesn't matter if it's the Y or the X-axis, but look here to make sure that each of your parameter names that are kind of indicated here by the comp and then the fluorochrome, right? Make sure that there's a double colon, and then after the double colon, there's a stain name after it. So for example, my Alexa 700 is attached to CD3, Pack Blue is associated with CD8, so on and so forth, okay? I have a color here, AARD, that doesn't have a designation. Now, if you guys didn't know what AARD was, and I gave you my data to analyze, right? How the heck would you know what that is unless you'd have to probably Google it to figure out that it's a viability dye, unless you've already used it, right? But what about all the other colors? If I didn't tag Alexa 700 as CD3, you'd be calling me up or emailing me to figure out what all of these colors correspond to. Okay, so you can tag the stain name to the file on the machine, which I think is the best way to do it, because that will carry over the information to Flojo after the fact, and you don't have to change anything in Flojo. Okay, but if you forget, like in this case, we forgot the viability dye designation here, you can always change it in Flojo. So that's what I'm going to do. I'm going to make an amendment to the naming here for the AARD parameter. Okay, so how do we do that? Okay, this is changing the parameter list name. So the way we would do that is go to the Configure tab at the top of our workspace, and then head over to the Edit Columns button, which is in the Settings band. Okay, so you'll see this Edit Columns. You click on that, it opens up the Edit Columns window. And then if you scroll down a little bit here, you'll see a whole bunch of stuff start to be labeled reagent. And there's usually like a thin gray line at the top and bottom of this section. So where you start to see this reagent, these are your fluorochromes, right? This is what you are seeing in that drop-down menu in your graph window. So select the one that you want to label. In our case, we want to label AARD, and we say add column here. That will add that to the panel on the right. Then you say OK. Okay. Then here, you get the column that allows you to type in a value, right? So you can see here the header of the column is AARD, and it's blank underneath. Well, this just happens to be our viability dye. Okay, so you can type in viability. Now, in order for it to show up on all of your samples, you need to copy this value to all of the other samples that you have in the list. I don't want to have to type this 36 times or 37 times. Okay, so what I'm going to do here is I'm actually going to make use of another tool that we have in the workspace, and we go to the workspace tab. You'll see this band called the keywords band. If you expand that out, you will see there's an option here that says copy value to group. Okay, when you do that, viability gets copied across. And now you go to any file, open it up, click the drop down, you see AARD, and then it says viability. Okay, so we know that that's our live dead marker. So tag your data, make sure that your fluorochromes are labeled so that later on when you create your plots and stuff, you know exactly what you are looking at. All right, so that's annotation sort of number one, getting the fluorochrome list annotated. The second type of annotation that we probably want to do is an annotation that describes something about our workspace, or sorry, not our workspace, but our experiment. So in my case here, you can see that I have a series of files that are labeled LD something or other. In my case, LD just stands for local donor. And that means it's essentially a patient number, right? Patient number 1, patient number 2, patient number 4, patient 12, et cetera. And each of these patients have a time course associated with them, but you cannot understand that time course from the file name itself, right? You just see NS and SPI, blah, blah, blah. So really what has happened here is there's a 0, a 20-minute, 1 hour, and a 2-hour time point. And I want to annotate the data so that I can, mix and match. I can sort by time point, etc. Okay, now, the easiest way to do that is to segregate the files first into a group so that you can annotate them accordingly. Okay, so I like to block highlight them like this. So all I did was, click on the first sample, then hold down the shift key and click again at the last sample that I want to include in the group. Right, that will highlight all the samples there. And then I'll just go to the workspace tab at the top, go to the groups band, and I will say group selected samples. And this is a quick and dirty way to create a group. So here I'm going to call this group, I don't know, my test group. Okay, and then that will take all of those files, dump them into this ledger. You can see that I have 19 files. Okay, and now, as I said, I want to annotate them. I want to put the time course information with these files. So the way to do that is to right click on the file itself. It doesn't matter which one, any one of them. And then you would say add keyword. And then here we can add some information, right? So I might call this something like time point. It's going to give you a new column header with a bunch of blank cells. Okay, and then you can fill in the values accordingly. Now, again, you can do this by hand, or you can make use of another handy dandy little tool here, this create keyword value series. I like this tool. It's kind of an advanced tool, but nonetheless, I'll show it to you guys because I think it's super handy for what we're going to do. So just make sure that the keyword that you're trying to annotate is the one that you just created, so that appears in this list here. Instead of being on numerical keys, you want to make this keywords, essentially text, and then type in the values as you want them to appear. So I want the 0 minute, the 20 minute, we'll keep it all in minutes. So the 1 hour and the 2 hour. This is essentially what I want. Okay, now, in this panel at the very bottom, you can see what the format is going to look like. Currently, it's going to look like 0, 20, 60, 120, and then it's going to go 5, 6, 7, 8, 9, etc. Okay, so it works for the first 4, but after that, the pattern kind of falls apart. So we have to actually change something about the rules box here. So the first one says fill 19 values starting from the current sample. Yeah, that sounds reasonable. We're going to leave that one alone. How about use the value 1 time before changing to the next value? That seems correct, right? 0 before you go to 20, then you go to 60, then you go to 120. That seems good, so leave that alone. Third one says start over 99 samples from the starting value. I would say no, we don't want to start over from the 99th sample. We want to start over from the 4th sample, right? So you say start over every 4th. Now you can see that the pattern sort of changes to something that you want, right? 0, 20, 60, 120, 0, 20, 60, 120, blah, blah, blah. You click OK, and it will add that pattern of keywords to the column here. Okay, so that's an easy way to sort of quickly add some annotation. Now what I can do is I can sort by the time point, right? Ascending, descending. If I want to sort by the patient number, I can just use the file name because it's arranged in, right, LD1, LD2, LD14 in numerical order that way, right? Reverse or forward. All right, now, last thing I wanted to show you, if we go back to the all samples, if you want to create a group in a smart way, if a lot of you are doing repetitive work, you want to create a template, you're going to dump, more samples into this workspace and have them analyzed later on, and you want to immediately segregate the samples into their functional groups, you can create a group based on some sort of metadata that already exists in the data file. So in our case here, our FMOs are labeled FMO, so I can create a group that traps FMO files by default. Right, so I create a group in this case by clicking on that bracketed plus. I can call this my FMO group. And then what I would do down here in the sample inclusion criteria box, you tell it what you want Flowjo to look out for. So in my case, I want to say, oh, please look at the file name and find any file that contains the text FMO in it. Okay, then you say create group and then close this and you'll see that you get a new group created, 7 files in it, and it has only your FMOs. The cool thing about this though now is if you save this workspace, that group will always look for FMOs. So if you save it as a workspace or you save it as a template, for all future analysis, all your FMOs, if they're labeled that way, are going to end up in this group. Okay, and if there happen to be gates and statistics associated with it, it's going to inherit those gates as well. Okay, so now that we have the group, let's go ahead and start with our FMOs. We'll start our analysis here. We can finally get on to looking at some images here and creating some gates. Your analysis is obviously going to be different than mine, so I'm not going to spend too much time on explaining, my gating hierarchy or my gating strategy. Your markers are going to be different from mine. You may be looking at different cells. Today, we're going to be taking a look at lymphocytic cells, and I'm going to double click on this population. You'll notice that the minute you create a gate, it gets this little I don't know, petri dish, they call it. Some other students have called it Yoshi egg sitting next to the actual gate name. And then from there, you can actually visualize that population and then, put some other parameters. So I like to start with a tissue specific gate. Then I might do, I don't know, maybe some doublet discrimination. You could do viability die, whatever. I'm going to do it in this particular order. And we'll call this fine. Single cells is fine. Double click on this. Then we're going to go ahead and look at our viability die. So again, AARD is viability. And then maybe put this on side scatter. We're going to go after the cells that are negative for the viability die, right? Those that exclude the die are going to be the live cells. Maybe something like this. And then from there, we're going to go ahead and look at lineage specific markers. So I'm going to separate my CD3s essentially from my antigen presenting cells. We want to isolate the CD3 positives here. Now, a lot of times in Flowjo, the data are going to look squished, right? Kind of like this. There's a lot of white space that isn't being used in a lot of different sectors on the graph window here. Positive space, right? And negative space in both directions. So to better visualize this, we can click the T button to rescale the data. Okay, so this is just going to change how the data looks. It doesn't change the actual values of the data. Okay, so what I'm going to do here is I'm going to click the T button and then I'm going to say customize axis. Okay, this will open up another window that has a histogram in it, but with a bunch of controls. Okay, so I can get rid of the positive space here fairly quickly by just clicking on the little minus button. You can also type in a value here if you want to. Okay, I like doing everything as fast as possible. You can move the width basis here to adjust the negative end of the graph. So you can zoom in around 0 if you want by extending this out furthest to the left. If you compress it all the other way, right, you compress that area around 0 because it's less visible. I like to stretch this guy out a bit. Maybe, just give it a little bit of space here off towards the left so you pull those events off the y-axis. And then you can click apply. Right, and that's going to stretch the population out quite a bit. I probably could get rid of some more of this positive space here. Okay, but we'll focus on the y-axis now. So click the T button, do the same thing, customize. A couple of clicks on the negative there, maybe stretch out, right, the 0, pull some events off of the y-axis. Then go ahead and click apply here and look at that, right? A lot easier now to isolate the CD3 positive cells away from those that are CD3 negative, okay? And I didn't do anything to change the actual data, I just changed the way it's visualized. And a lot of reviewers now will ask you to make sure that, all of the data are, visible in the plot here. So don't be surprised if a reviewer comes back and says, I want you to transform these data so that it is easier to see. All right, so I'm going to put some quad gates up to isolate the CD3s, but there's a billion different ways you can go. I'm going to take the CD3 positive, since we're going to be looking at T cells here, and then we're going to further split these along the CD4 CD8 axis. Okay, now this is an FMO, right? No CD4 is contained within this tube. So what this allows us to do is kind of create a gate that abuts the true negative, right? So this is a fluorescence -1 control, super useful, especially in multicolor experiments, right? Where you have smeared markers, or maybe you don't know what the marker expression is, anything that's not going to be clearly on off. I highly recommend using a FMO, and especially if you're new to your panel, always use FMOs so that you can judge where negative and positive happens to be. So we'll create that CD4 gate in that location. Now, as you can see, my hierarchy has gotten a little bit more complicated, right? We looked at it initially when we have lymphocytes, then we created the singlets gate, and then we did alive dead, et cetera, et cetera. Now, if we want to preview these gates, right, on other samples, we have to copy these gates first to the other samples before we can look at them, because you can see here in the upper right, the left and right arrow keys are grayed out. So the way to easiestly, the best way, in my opinion, to sort of apply these gates to the other samples in the group is to use this function. You right click on the highlighted population and you use this copy analysis to group. Okay, of all of the tools that I use in FlowJo, this is a shortcut that I use the most often because it saves me the most amount of time. So this is Command-Shift-G if you're on a Mac. If you're on a PC, this is Control-Shift-G. Okay, but it saves me a ton of time. You say copy analysis to group. It does the same thing as sort of dragging and dropping the data to the group. But all other samples now inherit those gates. Okay, and now when you click through, you can see where that CD4 gate happens to lie on all of the other samples. Okay. All right, now I'm going to go ahead and fast forward here since we're kind of tight on time. Let's just go ahead and create our CD8 gate on our FMO for CD4, okay? Because we know where CD8 is going to lie. We'll go ahead and create a CD8 gate there. All right, and again, now I have this new little gate added in my hierarchy, right? You can kind of see that the CD8 gate color is this black, sort of flat font type, and the gates that have been copied over have this sort of, I don't know, yellowish tinge to them, right? So the yellow tinged gates mean that they're all in the same position and they've been copied to the group. So all other samples, gates that have that same color means that the gate is in the same position as any other sample. If the gate happens to be black, it means that it's either a new gate or the gate has changed its position relative to the others. Okay, now you want to add this CD8 gate to all of the other samples. Same thing, right click, copy analysis to group. Okay, and then it'll get placed in the correct location in the hierarchy. So anytime you have to do some quick ad hoc analysis, the copy analysis to group function is super useful. Now, I should mention that the behavior of the gates, once you've copied the gates, pardon me, to the group, you can no longer delete them from the lower section here. So for example, if you move this CD4 gate off, you don't like the CD4 gate's position, you don't want to delete that gate entirely. All you have to do is highlight this gate, hit the delete key, and it'll snap it back to its original position. Okay, if you don't actually like the gate and you want to maybe create a gate that has more vertices or has a different shape, then what you can do here is you can delete it entirely from the upper portion of the groups, right? So here you have CD4 listed in the group pane. If you want to delete that, you hit the delete key and then you will get a prompt and you just tell it delete entirely and that will get rid of the CD4 gate. Okay, but you can't delete it from the lower section if it's been copied. Okay, but let's go back to our CD4 gate there. CD4 positive, kind of create a catch-all, and then let's go ahead and right click here, copy analysis to group. All right, so once we have those gates in place, we want to probably analyze the CD8s to see, because we have a lot of readout markers here for CD8 activity, right? We have perforin, phospho-ERK, we also have interferon gamma production. So let's look at these readout markers. So perforin, for example, okay, here is a nice smeared marker. Again, it's not easy to determine positive from negative, so I'm going to use the FMO for for this particular population. So what I'm going to do is I'm going to hunt around for that FMO. I'm just going to advance till I see the sample that says no perf. Okay, so I have CD8 on the y-axis, perforin along the x, and again, we can kind of create a gate here that's sort of a catch-all. I want to catch those really bright populations at the top. Okay, so you have a perforin gate. Right? We can find our perforin gate by locating that blue diamond. So that was the third icon that I had mentioned in the beginning. When the icon is fully filled in, it's just showing you which graph window is in the forefront. So we just created the perforin gate on this CD8 population from this particular file. Okay, so you can use that little blue diamond to find where you last created your gate. Okay, and then should I say copy analysis to group so that everybody else gets the perforin gate. Okay, and then we'll go do the same thing for like phospho-ERK. Again, another marker that isn't well separated from the negative. Good thing we have FMOs to sort of identify the positive and negative boundary. All right, so we're going to come here, do the same thing. I'm going to label this P-ERK, right? ERK is a MAP kinase that gets phosphorylated by another MAP kinase, right? Signaling molecule. All right, and then from phospho-ERK, we may change out on that same population, look at interferon. Okay, interferon is a little bit more on-off, but still, never a bad idea to have the FMO. You can't go wrong by having the FMO in your data set. Okay, so I always tell people, if you're even thinking about including FMOs, just include them because it's going to save you a lot of headache in the end. All right, so we create another catch-all gate there, do the same thing here, copy analysis to group, we're good to go. Okay, now what I've done here so far is I've isolated CD8 T cells that express interferon, phospho-ERK, or perforin. Okay, but what if I wanted to know if I had CD8s that expressed all three of these markers, or perhaps positive for two and negative for another, right? How do I arrive at those? Well, you can create very convoluted 2x2 gates, dragging these guys underneath one another to get to what you want. But there is a faster way of doing that. So again, I'm all about efficiency here. You want to highlight the gates that you want the combination for. So in our case here, we want everything that is a CD8 positive gate that expresses these markers. And we're going to use a Boolean tools to figure this out. So you head over to the tools tab, Boolean band. And again, you want to know, let's say, all the permutations, right? The individual Boolean functions are indicated here by the green cartoons, But if you want all the combinations, just click the combination gate function, click OK. And then what you're going to get is, right, your triple positive, triple negative, your, interferon positive, perforin positive, phosphoric negative, so on and so forth. Okay. And if you want to copy these gates to the rest of the samples, just make sure to copy the gates that have this sort of slash going through them. Okay. Otherwise, FlowJo will not calculate these gates properly if you do not bring them with their siblings here. Okay. So you can bring over, let's say these are the gates you're interested in. You bring them over along with their slash mark siblings. Okay, don't forget to copy these. You don't have to copy over all the and gates, all the permutations if you don't want, but just carry over these guys. Okay, so it's a fast tool to be able to figure out many permutations at once. I will say that it's rather taxing on your computer, so be careful the number of combinations that you want to handle at any one time. Close all other programs, make room in the computer's memory for the permutations that you are wanting to work with. Okay, so in our case here, I'm going to go ahead and delete these because again, we're not going to use them for the analysis, but I just thought I'd show them to you. All right, now, a lot of times when we have terminal populations like this, we want to add statistics. So I usually right click and say, add statistic. Okay, there's several different statistics here that you can choose from, but most of the time we're interested in MFI. So MFI is usually meant to be the median. Okay, you could also use the geo mean. These two are very good statistics for the MFI because they are resistant to the presence of outliers, right? So if you had a single outlier over here on this graph, the median or the geo mean is going to tell you that, well, let's look at a population here that actually has something in it. Okay, but if you had a single outlier that was out to 10 to the 5th, if you use the median or the geo mean for an MFI of interferon, it's going to tell you something around, I don't know, something around 10 to the 4th, where the bulk of the population happens to be. Okay, the mean, which is the average, is going to be heavily skewed by that single outlier. It's going to tell you that, the cells are actually out here when in reality they're further down the line. Okay, same thing with the negative populations. The mean is very sensitive when you plot data on a logarithmic scale. It's very sensitive to extremes. So we normally avoid using the mean in flow. Okay, usually the median or the geo mean. Now, you can see that in the workspace, there's already a statistic column. What is that statistic that it is reporting? That statistic is the frequency of the parent. So here, for example, the lymphocytes are roughly 88% of the total file. Okay? Of all the cells that were acquired, 88% or 87.7% of them were lymphocytes. The single cells, right, are 99.9% of the lymphocytic population, okay, not 99% of the total file. Okay, live cells, right, we have about 96% viability of the single cells. Okay, if you want the live cell statistic as a result of total or the lymphocytes, we have to choose a statistic from the list here. So I'll show you that in a moment. But let's start with the medians, right? The MFI. So I'm going to put the geo mean and the median here. My personal favorite is the median. But you just pick those statistics, make sure the population is interferon, and then pick the color that you want it associated with. So we want to go with interferon. Click add. You could, do it for any other. You could do it for any other population or color. So in this case, because I selected the live population last, FlowJo went ahead and added it there. But really what we want is we want to add these statistics to the interferon group. So you can always drag and drop those stats from a population that you made a mistake on to another file without any sort of penalty. Okay, and then I'm just going to delete them from the live cell population. Okay, now, we want the MFI for phospho, ERK, and perforin as well, but there we probably want the respective MFI's colors, right? So you want the geomean or the median for phospho-ERK along the Alexa 48 parameter. You're not really, probably not interested in interferon MFI there. Okay, and then we can do the same thing here. We can just swap out the population and say perforin, and then change the color here, and it'll add it to the perforin population. Okay, now, as I mentioned before, if you want to know what the live cell statistic is relative to the total file or relative to lymphocytes, what you can do is you can say frequency of blank, right? And then from here, where it says frequency of, you have this little drop down menu. You just tell it what you want. So if you want it of the total file, you can say that, say add. And again, because I have selected the wrong population in the workspace here, it added it to my perforin population. But here you can see that now it tells you the frequency of total, right? My perforin positive cells are roughly 5% of the total. Okay, but you can drag and drop this onto the live. You can see here that the live cells are roughly 84% of all the cells that you worked with or that you collected. Okay, if you want to know what it is of the lymphocytic population, again, you can just say frequency of blank and then say, I want to know what it is relative to the lymphocytes, right? Click add. And now it's going to tell you the frequency of the lymphocyte, right? Live lymphocytes, in other words, that you have is 96%. All right, so hopefully that's clear. We can take these statistics, right? Let's just take these and we can add them to our workspace by simply right-clicking and say copy analysis to group so that all other samples now have those statistics. Okay, and now it'll make it easy when we go to export our stats to the table editor. They're already there. We can just drag and drop them in and we get what we want. Okay, so it's about time now to make some comparisons between these samples. I personally think it's difficult to sort of scroll through each of your samples here and compare them one by one. So I like to use the layout editor. And we have to go to the layout editor anyway to create our figures for papers, right? So what I'm going to do here is I'm going to open up the layout editor by clicking on the L. I like to have a split screen so that I can kind of see what I'm doing. And then we're going to drag and drop populations from one location to the other. Now, all of my analysis so far has been done with the FMO group, right? I haven't added any of this analysis to my test samples, which is really where I want all of these gates to go. Okay, so the easiest way to transfer gates between groups is to actually just simply drag and drop them. You cannot do, unfortunately, a Control-Shift-G between groups. So we still have to, do the drag and drop in this case. You just want to make sure that when you drag it, you can kind of hit the target you're looking for that's highlighted in blue, you let go, and it will apply all of those gates now to the test samples. Okay, so now we can start dealing with stuff in the layout editor. Another quick tip, kind of an advanced tip, I like to hide my statistic nodes while I'm working in the layout editor. So I go to the configure tab and then I toggle the stat nodes off so that I can just focus on the gates. Okay, now normally we're going to focus on the terminal populations and see what has happened from, one time point to the next. When I bring these in, there's a couple of things that you can do here, right? You can change the iteration options, first of all, to go to sample. And then that way you can use these little arrows to kind of see what, happens from one sample to the next. And you can kind of witness all three changing at the same time. You can click on the drop-down menu and just advance to a given sample right away, right, to see what has changed. A lot of times you're going to want to bring in your ancestral populations as well. You have to describe, essentially how you arrived at the population you're looking at. So a couple of ways to do that as well. You can right click here and say, show me the ancestry on the plot. You get this nice little panel that has all the ancestry plots present, right? You can make this horizontal if you'd like, move it underneath, whatever. It's a pretty cool panel. The only problem here is that it's describing the path just to this population, and you cannot ungroup any of the, you can't ungroup any of the plots that are within this panel. So it's sort of limited. It's nice if you need to give a quick summary of what was done, but if you need to get more in detail, probably not your favorite choice. Now, for me, I hate having to move plots around like this, right? And then you're going to probably draw some lines between the plots, and then you want to say, oh, give me an arrow going forward. And then, I want to move this here and show that there, blah, 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 right? You're going to have a lot of stuff that you're going to point out via the ancestry. And you're going to also have organizational problems like this, where plots are not aligned properly. You're going to end up in this situation where you want to align top and maybe distribute them evenly, etc. This to me takes so much time. I really hate having to arrange my plots. So for me, what I used to do when I was in a grad school and when I was a postdoc, I used to use this tool called the grid tool. So you can see here it's sort of ambiguously placed amongst all the drawing tools, but towards the left-hand side, if you roll your mouse over, it'll say grid. You click on that, then click and drag in the layout editor, and it'll actually create a grid for you. And then you can actually expand the numbers of grids that you have. So if you want more rows or whatever, you just right click on it and you say, I don't know, insert row. You can add 1 or 2 or however many you want. Right, so now I have a 4x4 grid. Okay, so now what you can do is you can take your plots and you can drag them either directly into the grid, or my favorite option is to kind of put them off towards the side and then drag them into the sort of upper leftmost corner. And you can't really see it very clearly, but whichever plot you're sort of leading with, just make sure that you drop it over the left-hand corner of your grid, and it'll drop everything in there. Okay, and the nice thing is now you have all your plots arranged, at least they're spaced evenly and they're arranged right top to bottom. You can swap them in and out of the squares if you like. So rather than dragging them in all at once, you can drag them in kind of into the spots as you wish. And then if you want to get rid of the black grid around it, my favorite option here is just to tell it to not have a line. And in that way, your plots look nice and even. And again, here, what you can do is you can just use the iterate by options, right? Like iterate by sample, and you can see how these plots change, right? From sample to sample. Saves you a bit of RAM, gets you the image you want. If you want to see this across all samples sort of all at once, you can say create the batch report. I'll put this in a single column down. But batch report will eat up a little bit more of your RAM. You can kind of see here that Flojo is moving along a little bit slower. But what it's trying to do now is to produce all 13 of my plots here across this grid for each of the samples that I have, right? So now I'll get the batched layout. If I zoom in, right? These are all the plots for sample number 1. Then I get all the plots for sample number 2, sample number 3, blah, 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 until you get to the end. Okay, so that's another way of sort of using the layout editor to visualize all of the data. Now, you have to keep in mind, if you batch a lot of things, the layout editor is live, even with the batched outputs. So for example, if I come back and I move the lymphocytic gate off, you can do some backgating that way, and you can see how does that gate influence all the other downstream populations. Okay, so you can see here what's happening. So the fact that this is live, it chews up a little bit of RAM. And again, if you want to snap that gate back to where it was before, just hit the delete key when you select it down here, and it'll snap it back into the original position. All right, I'm going to delete that layout because we don't need it. Come back to this one, or maybe create a new one because, again, I want to show you some comparisons. So this stuff is cool if you're just looking kind of one-to-one. A lot of times you probably will make a figure from this showing the ancestry or something. But really what you want to do is you want to show, you know, what is happening with the time course here. How are these files changing? Are the cells actually stimulated and producing interferon or phospho-ERK or perforin as a result of the stimulus that's been applied over the time course? And the way to do that is to create an overlay. And so normally what I'm going to do here is I bring my terminal populations out, and then I can just go and overlay them with their subsequent files. So for example, here I've gone ahead and just dragged in the second sample, right, which corresponds to the 20-minute time point, right? Then I can do the same thing with the third one, right? And then we can do the same thing with the fourth. So we can kind of see with the third and the fourth, they look like there's some interferon action happening, right? What about with phospho-ERK and with perforin? Now, the way I just created that overlay, I think is a little bit slow. A fast way to do this is to just highlight the gate, say select equivalent nodes. That will highlight all the phospho-ERKs, and then you can just drag them into the appropriate plot in the layout editor. Let me move perforin around just out of the way. In my opinion, it's just easier to clean this up, right, than it is to sort of scroll, drag, drop, scroll, drag, drop. So in the legend, you can block highlight everything that you don't want, everything that's not patient number 1, for example. And you can see it clearly here now, right? All the patients that are not patient 1, we right click and say remove layer. And that will create our nice little overlay that we want. If you use my method, you'll get dual representation of one of the files. So we can just say remove layer. Okay, and we'll do the same thing for perforin. We can go here, we can say select equivalent nodes. If you don't want that duplicate, you can sort of deselect that first sample and then drag them in this way. And then over here, we can go ahead and again, highlight everything you don't want and say remove layer and put that above your perforin. Okay, if you want to include the time point in the legend, you can see here that it just has my sample name, the subset that we're looking at, and the count. You can change the legend so that it includes more information. So I want to include the time point. Time point is a keyword, so we click on the little key button down here and scroll down to the bottom where you find your time point. Go ahead and click OK. And you click OK here, and it'll actually add it to the legend. In this case, it's going to add it to the legend for whatchamacallit. You're going to add it to the legend for interferon. Okay, maybe for the other plots that are sort of, sitting next door to our interferon, because these legends sort of overlap with one another, maybe what we can do here is remove some of this stuff. So maybe we don't need the sample name, right? We just need the subset name and perhaps the time point, right? Because we know that everything that's in a line here is going to be the same, the same sample. Okay, so what I could do is just move this maybe out like this. See what I mean? This type of stuff just irritates me, and because it takes forever to get everything in order, right? And the sizes of things and whatnot. So hopefully in the future we'll have even better tools that make this a bit faster. But in any case, you can manipulate this. You can change the colors, and you can change the plot type. So in general, I probably don't want to look at these plots as dot plots. So let's just go ahead and quickly change them. You can select them all, double click, go to the specify tab here and change the dot plot out to be a histogram. I usually like to make the y-axis to be normalized to the mode so that the small populations are visible amongst the big ones. And then you can see you translate all of those populations immediately or all the graphs into histograms. And then we want to offset these histograms. So let's also right click here and say, I don't know, we can do like a full offset for this guy, maybe a half offset for the other one. And then my favorite option is a 3-dimensional view, right? Which is this guy that says, oh, stagger offset. Okay, something like this. Okay, and you can change the line types and stuff if you want to do that individually by just clicking in the legend. You can right click and say, okay, well, I want, you know, this guy not to be tinted, and all the others, I don't know, you can put dashed lines. I don't know, stuff like that, right? So anyway, once you have the aesthetics all set up and you have everything arranged and organized, now you want to see how this happens. How does this look for all your other patients, right? We can clearly see that the latter 2 time points for interferon are good for patient 1, but what happens with patient 2, et cetera? Okay, in order to get this to batch, you can see here, this is all grayed out, right? We want to make the report. So to do that, we should click on the iteration options here. And my advice to you guys is to use a panel iteration because it's the easiest. Okay, so panel iteration will temporarily blow up your, you know, graphs here, but a new box opens up, and the box is asking you basically how many samples you have overlaid with one another. Okay, so in our case, we have 4. Right, the 0, 20, 60, 120. Once you tick that on up to 4, FlowJo takes that data, and now it knows to chunk out the workspace into groups of 4. So it's basically going to take the first 4 files, overlay them, take the next 4 files, overlay them, blah, 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 all through the workspace. Now you can go ahead and create the batch report so that you can see how this, you know, how the other patients are responding. So here is patient 1, no big deal, because that's what, you know, we created. Let me make this sort of more full screen so that you guys can see the entirety of the graphs, right? Patient number 1, here's patient number 2, right? If I scroll down, we're going to get patient number 4, then we have patient number 12, and then we have patient 14. Now, patient 14 did not have a 2-hour time point, so we're missing that 4th file, but in any case, there are the data files or the reports. Okay, so everything that I've done so far, we have produced in you know, within FlowJo. The batch reports have turned out in FlowJo, but you don't have to do it to FlowJo. You can actually create a PDF for the batch report here if you want, so you have all of the outputs as a PDF. You can also do PowerPoint if you'd like. My favorite option here, though, is the printer, because the printer will give you 3 things all in one. You say printer, then you click the button that says create batch report. What this is going to do is it brings up your print spooler, and here you can, you know, put so many per row, so many per column, right? So you effectively can retile the data irrespective of the little dashed lines that FlowJo has here. And then when you go to print this, you can print the PDF, right? Or, sorry, you can save it as a PDF, or you can print the hard copy. It's entirely up to you, right? So you kind of get 3 things in one, right? The hard copy for your notebook, although I don't know that people use manually written notebooks anymore. And then there's the PDF, right, which is your nice file for figures. And then, of course, you can retile the data. So 1, 2, 3 from that simple menu option. The other thing you can do is you can say, oh, you know what? I don't really want to deal with patient number 1. I really think that, I don't know, patient number 12 is the optimal patient. I want to show this patient set of graphs for my figure. You can select patient 12 from the staging area that you set up here. Then you can go to File and then just say Export Image, and you can export this as a TIFF, an SVG, or a PDF. Okay. So hopefully that answers your questions about, you know, the export of these items. I can always answer these in more detail later, but again, we're running up against the clock here, and I really want to get to compensation. So we're going to go ahead and exit out of the layout editor. And the next thing I'm going to show you is the table editor. This one is going to go by real fast, so that's nice. So the table editor, we usually have to toggle our statistics back on because that's what we're going to drag into the table editor. So we'll click on this little screen looking guy. It's going to open up another module. And then just like the layout editor, you can drag in the statistics, right? You can drag in populations as well. So we'll do that with like perforin, for example. Okay, but I just want you to note that when we drag in perforin, you'll see that it always defaults to frequency of parent. So if you wanted some other statistic associated with perforin, right? Let's say not the MFI of perforin, but maybe you wanted the count or something, I would recommend that you actually add the count statistic here. So you just say count, right, from the list. Make sure it's on the perforin. The color doesn't matter. Right? You just have the count there, and then you probably want to copy that to the group, and then just drag in the count statistic, right? You only have to do that once. And then let's drop the frequency of the parent because you're probably not interested in that, and click OK. Now, if you want to make this look pretty slick, especially with respect to your MFIs, you can go ahead and highlight these and maybe heat map them if you want. So select the files that you want to have heat mapped in the table. Click the heat map button after you select them. If you want to add the keyword that describes the time point to better visualize the table, click on the edit button here or the edit tab and then say add column. And then just click keyword in the center. Pretty straightforward. Go to time point, click OK. And that's going to add your time point here. And you can move this, wherever you want. You can put it at the bottom of the list, in the middle of the list, whatever helps you visualize the data better in the end. But then click the little cog here in the upper right, and you're going to get your table. Okay, so there's your time point, right? Describes all the times. And then you have your heat map for everything. So it looks like here with respect to the MFI of interferon, you can see that just what we saw in the graphs carries over, right? The latter two time points are high. First two time points, nothing is going on. The pattern looks very different though for phospho-ERK, right? The 20-minute time point is high, whereas the 2 hour and the 1 hour a little bit, their signal begins to dampen. And then perforin's kind of all over the place, right? Perforin really doesn't seem influenced by the stimulus or the time course. It changes more by the patient than it does anything else. Okay, but you can rearrange the table here if you want, right? By time point, by patient number, even by statistic if you want, right? See what's hot, what's not. And then of course you can save this. So you can say file, save as, text CSV is probably the best option. You can also save as Excel. I recommend that even if you're using Excel, save it as a CSV. Excel can read CSV files just fine. So I would save it this way. That way you don't run into any strange symbols and having problems with your pipelines downstream. So save it as a CSV to avoid any headaches in Excel. The other thing that you can do with this table is if you want to put it next to your graphs that you have in the layout editor, you can come here to the table editor tab and then just change the display here to current layout. Create the table. You get a warning here telling you that the table is not live, but you can set it amongst your plots. So whichever layout you had open, that's where Flojo will batch that table to, right? So again, you're going to get that warning, and then you can park your table, wherever you want. Here, I dragged it mistakenly into the grid. Obviously, not the right place for it, but in any case, you get the idea. All right, so we're almost done. And the last thing that I wanted to talk to you guys about was compensation. We talked about it in the beginning in terms of like what was good, what was bad. But I just wanted to draw your attention to the fact that you can run compensation in Flojo if you want. So, you can acquire your single stain controls on the machine, and then later you can run your compensation in FlowJo. The nice thing about running compensation in FlowJo is that we have several different ways to compensate the data. There are several compensation algorithms that you can take advantage of in FlowJo that you may not have the same ability to on your cytometer. And I'll talk about these in a second. Okay, there's nothing wrong with doing the compensation on the machine. A lot of times, in fact, doing it on the machine is going to be a lot faster and a lot more convenient. So, you know, do it there. If you have any questions, just make sure that you keep your single stain controls with you. And then if you want to try one of the algorithms in FlowJo, you know, by all means you can. So we'll go to the compensation group here. And in order to, you know, we just want to make sure that all of our single stain controls are included here. So I have them, you know, if there's any other files from another group that need to be included, just make sure to drag and drop them into this group if they're not appropriately named. Okay, but in this case, they're all here. Then you click on this rainbow M at the top. This is going to open up the compensation wizard. And basically all this tool is a new window that has a bunch of like tables in it. So the top table, right, this always happens with Zoom, like getting this bar to move down takes a couple of tries. But in any case, the top table just has all your fluorochromes listed on the left, and then the single stain control that those fluorochromes should be associated with is kind of labeled in the sample column, right? So AARD, the fluorochrome, right, should be matched up with the single stain control for AARD, and it happens to be, so this is good. You get a nice little green dot. If there's a mismatch in the name, you get a yellow dot, and sometimes if FlowJo cannot find the correct file, you'll get a red dot. Maybe sometimes if FlowJo can't create the proper gates on the file, you'll get a red dot. I normally tell people don't pay too much attention to the color here. Just make sure that the colors, the fluorochromes, are matched up to the appropriate single stain controls in these first two columns. Okay, so I normally check one by one here, Alexa 488, blah, blah, blah. Here's Alexa 647, and it's putting unstained beads. All right, in this case, in this experiment, we had an extra detector left open, Alexa 647. And what we want to do is we want to just dump this from the compensation matrix because this detector, we didn't have Alexa 647 being run. So we don't need it for this particular setup. So we'll go ahead and just say remove this parameter, and it'll shrink the list by one, and it'll get rid of Alexa 647 being calculated in the compensation. Okay. Alexa 700 is matched up, PE, blah, blah, blah. All these guys look to be matched up just fine. Okay. Now, in the center, there's this comp name. You can name this whatever you want. If you want to give it a different name, you can type it in here, and it'll apply it to the to the drop-down menu in your graph window. Normally, I ignore this. I just use the mechanism I showed you in the beginning to tag the stain names. Okay. Now for negative and positive, so. In traditional or conventional flow cytometry, which is what most of you are used to that are not using an Aurora, for example, for your acquisition. Most other cytometers that are not Auroras use what we call traditional or conventional spillover calculations for compensation. Okay? And in that methodology, you have to define a negative population and a positive population. Okay, so the positive population is usually derived from the single stain control. There's usually a cleanup gate. So FlowJo creates a cleanup gate called size, and then it creates a gate on the histogram for only the positive events. Okay. The negative isn't always derived from the positively stained tube. A lot of times what FlowJo is looking for here are unstained samples. Okay, so unstained cells, a lot of times if it finds unstained cells or beads, it'll go ahead and use that as the universal negative and apply that to all samples here. Okay, if there is no unstained, then it tries to divvy up your single stain tube into positive and negative, and derive things from the same place. Okay, in my case, I have mostly beads for my compensation. You can see here that a lot of the graphs on the right, you can kind of see beads, right? The beads usually have more discrete, smaller, smaller, tighter populations than cells do, which are much more spread out and variable. Okay, so maybe I want to use, in this case, I don't know, unstained beads as my universal negative. So I can come over here, I can go to the cleanup gate for unstained beads that FlowJo has already created, and I can simply just drag that onto the negative column, and it'll now swap out beads, unstained beads for the negative. Okay, now that's going to be appropriate for most of my, single stain controls, but it's not going to be appropriate for all of them. Okay, because some of them, like the viability dye here, I actually am using cells for. So we're going to have to fix this in a second. Okay, but that's what the negative and the positive columns are telling you. Now, those are useful in conventional cytometry, conventional compensation, and when you do spectral unmixing. So if you guys use an Aurora, you can tick the spectral box here, okay? And you'll probably want to tick the optimize weights box after you're done with your gating here, but you tick the spectral box and you go ahead and add your negative and positive gates, adjust them as necessary. When you're done, you can click optimize weights and you can get your spectrally unmixed compensation matrix, okay? So method number one is to do a non-spectral compensation. So if you don't have an Aurora or a Symphony A5, I'm trying to think of, Sony also has a machine that is a spectrally enabled machine. But things like the Cytoflex or Attune, Novocyte, those types of cytometers are conventional compensation cytometers. So they will not be able to take advantage of the spectral comp options. But the Aurora, the Symphony, and the Sony machines can use spectral unmixing. So you can tick the box here and take advantage of that if you want to do it. Okay, now the other option that you have for a spillover algorithm is the auto spill or auto spread. And I'll talk about this because I think this method is really powerful, really useful, and can speed up your work quite a bit. And I'll show you how to use that in a moment, okay? But it's worthwhile to spend a little bit of time on the traditional comp just to show you how to do it in FlowJo so that you can get a comp matrix created sort of the old-fashioned way, if you will, in FlowJo. Okay, so we're going to use a traditional method, non-spectral in this case, because again, we ran these on an LSR2, which is not a spectrally enabled machine. Usually it has, one detector for every color that you run on the machine. All right, so from here, what we want to do is all of our stuff is matched up. Now we want to go to the lower section and look at the graphs. Okay, so for the graph section, we want to make sure that, in this case, we're looking at AARD. Okay, we want to make sure that the graph on the left and the graph on the right are the same species, right? So we have beads over here, cells over here. We have to consider the marker that we're looking at. In this case, it is AARD, the live dead, right? The viability dye. So since this is the viability dye and the single stain control that is positive is done on cells, we need to use unstained cells for the negative. And I highly recommend using unstained cells. Do not gate on cells that are negative that have the AARD stain applied to them, because even the live cells have some AARD attached. So it's always best to use an unstained sample here, okay? So for the negative, for AARD, we want to swap this out and we want to use unstained cells. Okay, so when we click the negative column here, we select unstained cells. You can see that it's using the size gate, which is fine. if we actually look at the overlay between the two gates, so the gate on the left corresponds to the cleanup gate that's applied to the negative. The gate on the right, or the population on the right, corresponds to the cleanup gate that's on this positively stained single stain control. And then what you have in the middle is the overlay of the cells that are actually falling inside the size gate on either side. Okay, so you can see here that the blue histogram, which corresponds to the negative, is overlapping quite significantly with the green histogram, which is being derived from the positive. Okay, so there's essentially no separation between positive and negative here. This would cause problems for us in our compensation because it looks like there is nothing that is AARD positive. Now, in order to get some AARD positive cells, we can do a couple of things. We can simply move the gate that we have on these cells into an area where we expect to find dead cells, right? So the cleanup gate is currently on a nice, healthy population of lymphocytes, right, as judged by forward and side scatter. But dead cells, as we know, are probably in the 10:30, 11 o'clock position over here. They're a little bit smaller and usually have more granularity, right? They're broken up a bit more. They don't look as clean and nice as your healthy cells. So if we move that gate into that position, you can see immediately that we find some really bright AARD cells. Okay, now to satisfy the rule where the autofluorescence background of the positive and negative should be the same, we should do the same thing for the negative, right? Because you can see here that even though the unstained, or the cells that are sort of negative for AARD, you can see that they still have an autofluorescence in this channel that is separate from the autofluorescence of the unstained. Okay, ideally in compensation, we want these autofluorescences to be equal so that they drop out of the compensation equation. So we're going to move this negative into the same location. And as you see, the minute I move it off into an area where there's some dead cells, you can see that that autofluorescence increases. Now we have overlap between the autofluorescence of the positive stain and the autofluorescence of the negative. Okay, so that makes a good compensation control here. And we have good separation between the positive and the negative. I'm just going to move that gate over off towards the right to include the positive cells in case there's any impact on that median. Okay, and then we can move on to the next sample. Okay, so for this one, this is APCH7. We have beads on the right, beads on the left. Everything is good. Now, normally, because we're a little bit strict on time, I'm not going to do this for every sample, but it's a wise idea to just incorporate one bead population. Don't grab both of these beads, because the second bead is going to give you another bump, and it's going to affect the median. But in this experiment, for the most part, it should be fine where the gate happens to be placed. Okay, now that's fine. Now, when I made that gate adjustment, though, Flojo has this annoying little bug that kind of switches this guy back to the cells. But what we really want here are unstained beads, how it was before. Okay, so beads, right? The autofluorescence of beads should be the same for the unstained bead as well as the positive stained bead, so that's fine. Okay, then we go to Alexis 488. This looks good. Again, I would put this on just one bead population rather than two. You can kind of see the little blip here, likely caused by the second bead population. We'll skip over it for the time being just to save time. All right, here's another situation where we have Alexa 700, right? We've got cells on the right, beads on the left. So what we want to do for the negative for Alexa 700 is we want to incorporate unstained cells. Okay, now the trouble that we have here is we want to use the gate that we have on the positive. We want to use that for the negative as well, but we don't want to touch the gate that's already present here. Okay, because this gate is in the right position for the dead cells. So we need to create a new gate on this lymphocytic population here. and use that as the negative for this particular color. Okay, so the way to do that is just to simply find the positive, right? In this case, let's just see our CD3 Alexa 700. There's the cleanup gate. That's what we want to transfer onto the negative, right, or onto the unstained. But if you can see here, the unstained samples, the cleanup gate is also called size. So if I drag and drop this on top of this guy, it's just going to replace it. So an advanced trick here is just to simply rename the gate to something else like cells, and then you can drag and drop that onto your unstained sample, right? And now you have both gates represented, which is nice. One for the dead cells, one for the Alexa 700 compensation, right? Again, because I want to match autofluorescences of the positive and negative. I want to make sure that the gate on the negative is in the same place as it is for the positive. So I come here to Alexa 700, follow this across, and I'm just going to make sure that this guy is using unstained cells, the cells gate. Okay, and now I know that it's using this bottom one. And I have to make an adjustment to the overlay in the center. For whatever reason, FlowJo's peak finder, I don't know why, but it focuses and it picks up the negative population here, probably because it's contiguous with the positive. So it doesn't quite know where to put the cutoff. But just adjust that gate so that it's in the right position. And that will take care of that. And I think all the other colors are okay. We have beads matched with beads on both sides, right? Beads in the positive, beads on the negative. And we're good to go. Okay, so when you're ready, this is like your traditional, we could just say traditional comp, something like that. Say view matrix, it's going to go ahead and create a new compensation matrix for you in the kind of the matrix editor here. So if I zoom out, right, you can see now we have this traditional comp matrix on the left, right? The one that was previous, was this one, right? But now we just created this guy. And if you want, you can actually see how well you did versus what you did on the machine. You can actually highlight both of these guys by holding down the control key or the command key. And now you can see how the blue dot, or sorry, the red dots and the gray dots overlap with one another. Right? You can kind of see here that, yeah, for the most part, they're superimposable. There's very little, subtle changes in the positions of the red cells relative to the gray cells. So this basically means that both compensation matrices are equivalent to one another. And if you look at the panel that we have above here, the largest difference is in AARD, right? We have almost a 1 point difference in one matrix versus the other, but when we actually output this, that 0.8% difference doesn't really impact the data significantly. Okay, so we have equivalent matrices essentially in both. Okay, so that's traditional compensation. If you want to apply this matrix, let's say you like it better than the other one, all you have to do here is click and drag the M button and then drop it onto the group of choice. So this always happens with Zoom. I can't always drag this on the first try or even the millionth try, so I'm going to leave that alone, but you can use this function here to just say apply to group. And now the FMO group is going to have that new red matrix that we created. All right, so I want to show you guys the really cool trick here, though, and that trick is to do auto spill. So auto spill uses a linear regression algorithm rather than matrix algebra to determine the spillover coefficients of how much dye spills over into any given detector. Okay, so it uses linear regression, and therefore it likes to see spread in the data. So this particular method is useful, particularly useful when you use cells as your compensation controls. So for this guy, the setup is actually much simpler than the traditional or conventional compensation mechanism. Okay, so the first thing you need to do is just tick this over to auto spill, auto spread. Then you want to say remove universal negative. So this does not actually use a negative positive designation. It basically needs a cleanup gate, right, where you focus on some cells that hopefully have some expression, some widespread expression of the marker. So in this case, where the AARD gate is perfect because we have nice spread in the data. Okay, for the beads, there's really not much we can do here. Those beads are just pretty much on off. So the regression curve in this case is going to have a bunch of data points on the negative end, a bunch of data points on the positive end, and that's about it. And these regression lines are going to try to move the, the median of the single positive population. They're going to try to move that median down so that it matches the median of the, essentially of the negative population, or the negative end of the graph. Okay, because there's no negative that's truly designated here. It's just going to look, say which point is the furthest left. We're going to try and make that median the lowest point, and then we're going to adjust the regression curve accordingly as best we can with the data points that are presented to us. Okay, so auto spill, universal negative. Now you just go through the data to make sure you've got any spread if possible. Okay, not much, as I said, we can do about beads. Lymphocytic gate has nice spread. We got positive and negative present and some stuff in between. Okay, that's essentially all we have. And then what we'll do here is we'll name this auto spill underscore with no AF. So the other thing that auto spill can do is it can subtract out autofluorescence. So if you have highly autofluorescent cells, you may want to choose this methodology for correcting for that autofluorescence. So I'll show you how to do that momentarily, but then we can say view matrix here, right? Look at how quickly we generated a compensation matrix without having to adjust any positive, negative gates or anything, right? We just created it, right? Clicked a couple of buttons and it created a new matrix for us. Now, again, if you want to compare this to your traditional matrix, you can see here there are some very small differences in the values, but when you look at the actual plots themselves, the blue dots and the red dots, again, are virtually superimposable with each other, right? So there's no big shifts of the cells in one direction or the other. So for a lot less effort, we generated effectively the same compensation matrix, which I find to be super cool. Now, as I mentioned, you can get rid of autofluorescence here if you have an extra channel that you dedicate to those autofluorescent cells. So in our case, it just happens to be that we had that blank channel, right? The Alexa 647 channel. So we could use Alexa 647 here to define the autofluorescence. But I'll tell you guys, if you're going to use this in the future, treat your autofluorescent cells as part of your panel. So if you know that the cells are autofluorescent around 550 nanometers, then make sure that that detector that's in that range is left open. Don't put a color there. In this case, a lot of times the cells are actually autofluorescent around 650 nanometers. So the Alexa 647 channel is actually pretty perfect for that purpose. So what you would do is you would go to your unstained cells and then just draw a gate on the autofluorescent population, or the one you think is autofluorescent. We can kind of draw a gate here. We'll just call these autofluorescent, and then you plug the autofluorescent guys into the positive here for that particular channel. Again, we're going to remove universal negative, check our gates on our samples. So ARD here, we got to move that gate back over into an area that we find some dead cells and some spread in the data. Okay, but again, just clear this. So we got spread, we have our beads, which we can't do anything about. Now, unfortunately, my cells are not too autofluorescent, so this isn't going to show you a great result, but this is how you would actually remove or subtract out the autofluorescence. Okay, you just plug that autofluorescent population in to the channel that you had left open. Okay, and then you just say, this is going to be auto spill underscore with AF removal. All right, and you click view matrix. It's going to take a few seconds here, go through a few iterations, and you'll usually see this line in your table that has a lot of red. If you actually look at the autofluorescence though, and we go to that channel, right, here's your Alexa 647, you're looking at the autofluorescence, right? You can see some cells here tend to be fairly bright. There's not too many of them, but when you do the autofluorescence subtraction, you can see that it flattens out those cells, right? Makes them more parallel to the axis. And in here, you kind of see that spreading. Maybe it was a little bit aggressive on this side, but not terrible. Okay, so in any case, look, you created, again, just a virtually equivalent matrix to the others with autofluorescence subtraction and very little effort. So I highly recommend that you guys check that out. The paper that was published in 2018, Carlos Roca was the primary author of the paper. So that's ROCA. And I don't know, I would just check it out. It's a really great paper. And I have to say that the vast majority of data sets I've worked with so far seem to work really well with this auto spill. And it's half the work. It's probably 1/10 the work that you're used to. Okay, so that's essentially all I had for you guys. Saving your work is pretty straightforward. Click the file, save as. You can save it as a workspace or save it as an ACS or as a template. The point is the difference between these different formats is whether or not they include the files. So the workspace does not include the files. It doesn't embed them. It just remembers the location of where your FCS files are on your hard drive. Okay, ACS will embed the files into the workspace, so it's kind of nice for sharing. And then the template forgets everything about the files, but retains the gates, the statistics, etc. Okay. So if you guys have any questions, I'd be happy to take them. If not, by all means, you guys can send me an email, jack.flojo@bd.com. And I'll wait around. Yes. Oh, hi, Bridget. Yeah. How are you doing?
Hey, I'm good. I just wanted to ask if you could briefly plop into the cell cycle platforms. There is one person that may be on the list today and joining us, or may check the recording, but was interested in looking at cell cycle measurements and S phase detection.
Ah, okay. Yeah. I can just.
Now they aren't using BRDU. They're not using BRDU. They're just looking at yeast and some pretty interesting profiles in cell cycle work. And I think, I've directed them to the online tools that Flojo has, but just so we have it in the recording to just show them where those features live.
Oh yeah, sure. Let me just bring in, I have a cell cycle data set, and I'll just bring that in real briefly and then I can open it up. So we have cell cycle. I think I had 2 different experiments, but there they are. Okay, cool. Yeah, so for cell cycle, just drag in your samples, go ahead and create, gates as you normally would on, on the cells that you're interested in looking at. So for example, here I have some cells and I'll copy that across to the group. And let's see, just to make sure that the cells gate, yeah, it's across all of them. Now, normally what I will do is if you had, you're going to use like DAPI or some sort of DNA dye, Flojo doesn't really, calculate it doesn't calculate like a second S phase or the 8N population or the 16N population away from each other. So it's kind of up to you how clean you want the model. If you only want to pay attention to the euploid populations, you can create a gate on the euploid cells, and then just run the model on that. You can run the model on everything like we're going to do here. You can clearly see that there's some cells here that are definitely polyploid, and you'll see how the model sort of builds them in. But you just basically create a cleanup gate to get rid of the debris. I'd start on a nice control that has hopefully has well-defined positive G2 peaks and G1 peaks, right? So if you use cell lines, this is easily seen. If they're random cycling, you can easily see a positive and a negative. But if you're using primary cells, I highly recommend that you use controls that use drugs to inhibit the cells in either G1 or G2 so that you can identify them quite clearly. Usually primary cells have a hard time finding the G2 peak. So a little bit of nicotazole or what's the other one? I always forget this, the tubulin inhibiting colchicine. So either nicotazole or colchicine will lock cells in the to find the G2 positive cells. So that's what I would use. But if you have random cycling cells like this, it's easy enough to to create the gates on the G1 and G2 peaks, right? Normally, what I would do here is I expand the models and the constraints that you have down here. So the model, it's up to you whether you want to use Watson or DeanJet Fox. These are published, one is like 79 and the other one is like 80 or 81. So Watson was first, DeanJet Fox was later. The difference between the two is that Watson uses a subtractive model. So it just takes, it adds G1 to G2 and subtracts it from the total. So anything that's underneath this pink curve, it basically subtracts out. And then whatever's left over, it says is the S phase, which is kind of this yellowish population that you see in the middle. There's a question here that says, any way to identify 8N and 16 cells? No, not proper. So what you'll get here in the stats band is it'll show you here that you have greater than G2, you have roughly 9% of cells are greater than G2, but it will not model them for you. So if you wanted to model your 8N and 16N, there's a couple of ways you could do it. You could just gate out, for example, your euploid cells. Right, so let's go here and you could say, okay, well, I'm only interested in gating the euploids first, right? So you'd say euploid, and then your larger than 8N, 16N, right? You could call these your polyploids. And then what you could do is you could try to adjust the cell cycle platform underneath, use it underneath this population, and try to adjust it to this so that you make this your G1, right, and this your G2. But the odds are, if you don't have enough cells in these locations, you're not going to get a really good model. And the reason, what I can tell you here is, you can look at you can kind of see these squarish sort of Minecraft-like projections within the model and the data itself. And this is telling you that you have very few cells here. So unless you have a lot of cells that are 8N and 16N, I wouldn't recommend adjusting the model to this. You can try it, but in my case here, these statistics are going to be very, very wild and swinging in a lot of different directions there. Okay. If we go back to our initial model here, I just basically set up the constraints using the random cycling cells, but I would probably, again, using primary cells, I might use set the constraint on G2 using that colchicine or nicotazole inhibited subset, and then coming back here to figure things out. So as I mentioned, Watson is subtractive. DeanJet Fox, instead of being subtractive, it actually fits a polynomial to the S phase. That can be useful if you're synchronizing cells and then you release them. So this will basically just cut up the S phase into 4 different equally, partitioned phases. But it's, in general, Watson I find to be, I don't know, well, they're both published, so they're both good. It's just a matter of sticking with one. Whatever you want to use for your analysis, stick with that and use that to report. I tend to find that DeanJet Fox is more flexible, obviously with the S phase because it's fitting a curve to it, versus Watson is a little bit more stringent. Once you set the constraints for the peak values, the next thing that you want to do here is I always set the G2 peak to whatever the G1 value happens to be. So this is kind of a critical, I think it's a critical step. Take note of the G1 peak here if you like it. So for example, we see that the G1 CV is 7 and the peak is at, I don't know, 192 or whatever. You can constrain the CV. So you can say, okay, well, this is 7. 7 looks pretty good. Then what I would do here is I would set your G1 to that value that you see here, which is 7. So you say constrain to n equals, and then we put 7, hit enter, it'll pop back. And now what you can do is you can say, make the G1 CV equal the G2 C, or make the G2 CV equal G1, or just add the 7 there so that they're the same, because the width of the G1 and the width of the G2 should be the same across your experiment. Okay, and yeah, so from there, I mean, that's how you would set up the model, and then you can apply this model to the rest of the samples by simply dragging and dropping that onto the gate that you want. And now you have a cell cycle platform that will be applied to all of your, even your ugly looking samples. And then you can derive all of those, you can derive all the respective statistics that you want from it. If you want to, export the statistics very easily, just drag the cell cycle platform into the table editor, and it'll give you all of the statistics that appear in this panel. Right, so whatever you have here, the RMSD, the G1 percentages, all of this stuff ends up in the table editor. And then of course you can create your you can create your panel or your table, and then you can whatchamacallit, you can export that to Excel. Okay, and the same thing in the layout editor, you just drag in the cell cycle node if you want, and you can overlay cell cycle nodes. So I'm not sure how useful that is, but you could, if you wanted to, just kind of show the differences between, maybe you've got more polyploid cells in one sample versus the other. Perhaps the S phases are significantly different, right? So maybe, overlay the control with a really ugly sample here. Maybe this B1, and you can kind of see, okay, yeah, the S phase in both is pretty different. So basically all the manipulations that you could do, with them as gates, you can do them here, okay? And if you have any other questions about the cell cycle platform, I'd be happy to answer them more specifically. So you can email me and we can do a little one-on-one if you prefer.
Thanks, Jack. That's really great. I appreciate that. Well,
sure, no problem.
I think this has been a really great overview of the features in FlowJo. Jack, I know you've got a lot of tools on the FlowJo website as well that are available, but maybe point folks in that direction. But remember that we've recorded this session today, and we'll direct you to that recording as soon as it compiles.
Yeah, certainly. The additional help can be found underneath the Learn tab. So if you just go to flowjo.com, you'll see Learn here. We have webinars that are upcoming that will be shown here. So today, for example, we had a great display of a new Seurat plugin that was just developed by Josh, and he gave the seminar today. Chris gave the intro to FlowJo on the 1st, but we should have some others coming up. So QC with Peacock is going to be done by Veronica on the 15th. And we have some other stuff popping up here, as you can see, in the coming months. Recorded webinars that we've done publicly for the world, if you will, are recorded and placed here. So you can see specific tools like Cluster Explorer. You can see a full high dimensional workflow. We used to have some stuff on cell cycle, but perhaps that'll be the new one because we haven't recorded one of those in a while. But in any case, they're all listed here, and you can see if there's a topic that you like. On top of that, there are FlowJo University short video tutorials. So you click on the program that you want to or that you are using, and it has some tips and tricks, a bunch of short little videos here that show you different different tools in FlowJo, and they're usually like, I don't know, 10-minute videos at most, maybe 15 minutes, etc. Okay, and then we also have tutorials as well. These they come with a printed manual. I don't know. I've heard from a lot of people that print is dead, but if you like print, you can download the printed manual here, and it actually comes with a data set. So it's kind of nice if you're brand new. You can use that data set to kind of plow through and work through the data and the instructions. Okay, and as I said, this has been recorded, so I will forward it as soon as it gets compiled to Bridget, and then she can disperse it that way. Otherwise, you guys can email me independently.
Super. Hey, I just want to thank you again, Jack, and thanks everybody who joined us today. Appreciate all of you taking the time. Please remember that same time next Wednesday, we will have an advanced session, and I've found that information to be extremely helpful for many of us that are doing multi-parameter flow cytometry. That's next Wednesday. We'll send out a reminder, and 10 a.m. to noon. Thanks again, Jack.
Cool. Yeah, no problem. Look forward to seeing you guys next week. Take care.
Cool.
Thank you, Jack.
All right. Take care, Jonathan.
Take care.